So let me just turn to the podium to Professor Manuel Llinas, true pioneer of malaria metabolomics.
Okay, good afternoon, everyone. It's a pleasure to be here. It's nice to see that metabolomics is increasingly attracting people's attention. The room is every year slightly fuller for these types of talks, which is really fantastic. So I think we have a really exciting lineup. I'm going to kick off today talking about the types of things that we've been doing in my lab, looking at plasmodium metabolomics over the last several years. So all of this is pretty much facilitated by the fact that, as has just been pointed out by Shuzhao, that technology is really advancing very rapidly. And what that allows us to do these days is really simultaneously measure hundreds to thousands of different metabolites in very complex samples, in part because of the combination of both the very sensitive detectors on the mass spec side with chromatography that separates things out, giving us multiple dimensions upon which to measure our data, including retention time and then the mass to charge ratios from these types of instruments. And of course, many of you are aware that these types of experiments are also often done using nuclear magnetic resonance. So there are two basic ways that we can ultimately analyze these types of data. These include targeted mode, which is a very streamlined method in which we have already run, at this time, about 250 well-defined metabolites through this analytical platform. We know exactly their retention times and mass charge ratios. Of course, when we're doing that, though, we gather all the full data. We ignore some of it in our analysis to make our lives easier, but there's much more data out there because, in reality, what we're measuring are thousands of different mass signatures in these types of experiments. And just to give you a sense of what we can capture, so many of you have seen this type of depiction before. This is essentially a metabolic a map of the plasmodium metabolism based on the genome from the original genome studies. And overlaid here are the metabolites that we essentially measure in a single experiment from any given sample of plasmodium parasites. These databases are increasing in size, so depending on what access you have and what instrumentation you have, obviously the numbers that you're measuring are variable. But the point is that we measure central metabolism very well in a diverse set of pathways that are obviously important to the parasite for its development. So some of the basic questions that my lab has been addressing are outlined here. Because this is a parasite, measuring host-parasite interactions, so nutrient exchange is clearly of importance because that is what allows the parasite to actually survive. We've gone on to do experiments where we're looking at actual metabolic flux through particular pathways within the parasite. We've published on trying to make connections between genetic differences in parasites and how that alters metabolism, or looking at metabolic differences and then asking what are the genotypic differences and actually making connections. This is some work that we did with Michael Fertig's group. Perhaps the least explored area so far, but where we have nice data is that, we see metabolites that are clearly being made by plasmodium parasites. And, that to me is a very exciting area because ultimately enzymes are making these things. And if we're thinking about future drug development, we'd like to have areas that are specific to the parasite that could ultimately be drugged. So what I want to talk about today, though, is something relatively new from my lab, and also you'll hear more on this from Darren Krieg's group, and that is looking at metabolic responses to anti-malarial compounds, in particular the malaria box. So we call this metabolite signature profiling, and the basic question is quite straightforward. Can we actually identify the cellular targets and predict mode of action of current and potential anti-malarial drugs? And then what do we actually do with this kind of information with regard to thinking about future anti-malarial drug strategies? And this work has been recently published both from Darren's lab as well as my lab in this journal here. Okay, so many of you will be aware that there have been a number of anti-malarial screens that have been done. This is obviously not comprehensive, but these are the screens that are ultimately contributing to what is being termed the malaria box. So greater than 5 million compounds have been tested in large-scale assays against Plasmodium falciparum. There's a very large number of hits, and they've been sourced from these different sources in this particular instance. And 400 of these compounds have been placed within this malaria box. This malaria box that was made available to the community to do a number of different assays. This box is supposed to represent a very large chemical diversity and different scaffolds, etc., but all of high potency against the parasite in the blood stage. So as I just reiterated, this box was made publicly available. Our mission in analyzing this box was to use mass spec-based metabolomics to measure actual metabolic changes in response to these drugs, and using both a targeted analysis and an untargeted analysis of the data that we generated. So I think this probably sounds relatively straightforward, but I do want to highlight that there are a number of different questions that could result from these type of experiments. So one, we would like to address: what are the actual pathways that are targeted directly by these anti-malarials? Can we use this information to actually determine the mode of action? If we can determine these two parameters, this could ultimately use to think about prioritization of lead anti-malarials. You can imagine also for combination therapies, because you want to, ideally, maybe we want to pair drugs that target different metabolic pathways. And lastly, we've been recently working with several groups trying to incorporate these types of data into better understanding resistance mechanisms in terms of how parasites ultimately overcome some of the pressures from these particular drugs. So there were a lot of parameters that we had to consider to set up this experiment. This is a medium throughput experiment at best, in large part because there are a number of possible parameters that you could modify to try to characterize a large number of drugs such as these using metabolomics. So quite simply, we need to think about concentration of drug, how long you actually expose the parasites to the drug, what do you ultimately measure? In other words, what mass spec type of platform are you going to use? And, maybe different parasites are actually going to respond differently. So these are all considerations. And so the data that we do generate needs to be taken with a grain of salt, because it is under very particular parameters for all of these different questions. And I'll address those in a moment. So as a proof of principle now, a number of years ago, we tested a well-known, a well-established drug, atovaquone, which is known to be essential in Plasmodium for pyrimidine biosynthesis. This was shown a number of years ago by Akil Vaidya's group, and essentially blocks the passage of electrons from ubiquinone to cytochrome BC1, and it's an extremely potent drug. So using this molecule in treating parasites, we would expect to find a blockage here in the pyrimidine biosynthetic pathway, and using mass spec-based metabolomics, we can focus on intermediates in this pathway. And as you might expect from classical biochemistry, you would expect a buildup of metabolites that are in this pathway if it's blocked at a particular point in the actual progression of the metabolic pathway here. So and that's exactly what we see. We see an increase in carbamyl aspartate, Ca here, and dihydroorotate that's demonstrated here in atovaquone. You could also target this pathway from a different angle. So in this case, we've used DSM-1, which now directly inhibits the dehydrogenase at this step. You would expect sort of the same metabolic response from those parasites, and that's what we're showing here. So we've used atovaquone as our sort of workhorse to define a lot of the parameters that I initially described to you needed to be tweaked to do these kinds of experiments. So the first thing you see here is determining which stage of the parasite to actually target using these anti-malarial drugs. You can see that there's a slight variation in which stage gives you the strongest signal. We ultimately would choose trophozoites out of these particular experimental parameters. Secondarily, there are a number of ways then to isolate parasites. So here we compare bulk extraction, saponin extraction, or magnetic purification of parasites before extraction of metabolites, and you can see we get a much more robust signal if we do magnetic purification. The third parameter we looked at is how long should we expose the parasites to the actual drug, and you see that there's a significant increase here when you move from 2 to 4 hours. So most of our experiments that I'm going to show you today have been done at 2.5 hour exposure of drug. And lastly, asking the question of how much drug should you actually use, in part because what we don't want to measure is just watching parasites die. We'd like to actually be capturing the response. So there's a very fine line with how long you expose and how much drug you actually put on the parasites to capture an actual response to the particular drug. And you can see that certainly 10x IC50 gives you a good response. You don't get much diminishing returns after you go to 20 to 50. So we've been working down in this range between 5 to 10x the IC50. So to answer these questions that I laid out before, all the experiments that I'll show you have been attempting to target 10x IC50 at the trophozoite stage, measuring steady-state metabolites using mass spec-based approaches after 2.5 hours using the 3.7 strain of Plasmodium falciparum. The methodology is outlined here. Essentially, so this is what makes it a medium throughput experiment. It's not that the mass spec holds us up. It's really all the culturing that has to go into doing this, because we require a fair bit of material to actually get very robust signals. We, of course, are working with highly synchronous cultures that we then, as I mentioned, magnetically enrich. So we get these really nicely enriched trophozoites. We then put them back actually in culture for several hours with media without drugs, so that they sort of restabilize if there's any perturbation due to the magnetic enrichment. We then expose them to drug, and we do the extraction, and then the metabolite extraction and mass spec analysis. So while this may sound laborious, the actual labor comes right here. Okay, most of our time is actually spent in analyzing data, not extracting peaks per se, but trying to make sense of what the data are actually telling us. So very early on experiment following on from atovaquone in this line of inquiry, we entered into a collaboration with Jeremy Burrows at the MMV. They sent us 11 advanced stage compounds from the MMV pipeline. We were blinded to what all these compounds actually were. We were just assigned numbers, and we were told we were assigned numbers, and we basically set out to predict and to report back to them what we thought the mode of action for these different compounds were. The way we are trying to visualize these data is using what we are terming metaprints. They're based on this R bioconductor package that is called a suprahex that was published not too long ago. What's nice about this form, it's a heat map, right? But it's condensed into these hexagons, where each hexagon within the hexagon actually represents different aspects of metabolism in your data set. So what you can see here, for example, is pyrimidine precursors here in red are up in one corner, and the other corner here is folate biosynthesis. And so if you add a drug like WR99210, which affects folate biosynthesis, you get a very strong signal in that particular corner of the hexagon. Conversely, if you look at atovaquone, now you get a signal over in this corner. And the reason we're using this is because it really gives a quicker way of comparing different drugs to each other to ask how similar or different are they from each other with regard to the parasite's response. Okay, so if you look at these 11 compounds that we tested, they fall into these, there's 5 categories here, but really 4 of them we can actually say something about with regard to what we think that they're actually doing. You have here a group of folate biosynthesis inhibitors, others that are in a group that we called cellular homeostasis. These end up being all of the PFATP4 targeting drugs. We have a set here that are involved that manifest in a hemoglobin catabolism type response. And then lastly, down here, perhaps the most obvious signal that we've seen, and that's molecules that are similar to atovaquone, again affecting pyrimidine biosynthesis. So I hope that you can appreciate that these types of patterns are actually a little bit simpler than displaying the data like this. It's the same data, okay, there's nothing different. It's just that the clustering is exactly the same, but I think the other format of looking at it is a little bit more intuitive. And of course, it's up to you how you want to actually visualize the data and think about it for your own benefit. Okay, so we've also then gone on to now report on a subset of the malaria box compounds. In our recent paper, we looked at 169 drug-like compounds. Of these, they fell into these broad categories that are outlined here. So again, this was a little bit surprising, I would say, given that the malaria box is stated to have a very broad or highly diverse chemical composition, presumably maybe hitting very diverse pathways. Maybe that's actually true, because if you notice here, 59% of these actually have an unclassified category, which I'll elaborate on in a minute. But overall, they really do fall into these four major categories that you see up here. Okay, so again, looking at all the data now, what we're trying to depict is that you see these four major categories, black being the unclassified region, and out here in red, atovaquone-like. You see here, these are molecules that affect pyrimidine biosynthesis. You see a very strong signal for these two metabolites that I told you about earlier. And down here are representative metaprints that actually represent those different categories and the drugs that are in each of these individual categories that we reported. If you look in particular at the green category, these are 23 malaria box compounds that we've designated as cellular homeostasis, in part because we don't quite understand what is actually going on in the cell when we actually expose them to these drugs, but they all respond very analogously to each other. Of these, 19 of the 23 have already been reported as PFATP4 inhibitors. So in other words, it's already known that they all function similarly, although when you look at what metabolic perturbation is happening, it's hard to reconcile that a particular metabolic pathway is being directly targeted by these actual compounds. So there's some broader manifestation going on, and hence our description or depiction here calling it cellular homeostasis. And as has been reported in the literature, of course, that's probably to be expected, given that this is, presumably due to the loss of sodium proton gradient and other downstream manifestations of a loss in that. Of particular interest probably moving forward is this group of 87 unclassified. These are in the black box. So there's lots of reasons why these things may not actually be giving us good signals. Of course, our methods, we could just be looking, we're using a particular metabolic mass spec platform. We aren't focusing on hydrophobic molecules. We're not looking at fatty acids. So there's many reasons why these things may still have metabolic effects, but we just didn't measure them in this particular experiment. The stage at which they actually have their effect could be different than the one we actually measured. The time of exposure, maybe some of these drugs are actually more long, they take longer to manifest and to give us a particular signature, and the 2.5 hours that we use is not significant. Maybe the dose of compound is actually too low. Maybe the drugs aren't as pure as they actually claim to be. And lastly, some of these compounds may actually just target broader aspects of parasite biology, and so you just sort of get a generic response, and maybe that's what a lot of that unclassified segment of our data actually represents. So we can monitor, we can change some of these parameters up here, and really that'll be done sort of on a case-by-case basis depending on prioritization. So where else would we like to go with this? Obviously, we're going to continue working with MMB and other entities to define what some of the pathways are that are being targeted by next generation antimalarials. But we'd also like to think about incorporating an untargeted approach. So I'll just give you one brief example of that. And we'd also like to move into other stages. Blocking sexual stage development is of great importance to our field, and yet we don't know much about the actual metabolism of the sexual stage development, nor what the sensitivity of that stage actually looks like. And some other things listed here, of course, we have already started extending our studies to look at metabolomics of whole mosquitoes. It's amazing that this actually works. If you just grind up mosquitoes, it's extremely reproducible, so some really interesting data emerging there. We should be looking at other species like Plasmodium vivax, and we're looking into samples from the field, and ultimately thinking about profiling infected individuals. So these are more long-term goals These are some of the things we've already started diving into. And here's an example of an untargeted molecule. So here's a mass. You can clearly see that when we drug with this particular compound here, we get a striking increase in this particular metabolite with no drug. There's almost none of this metabolite. What this is, I have no idea. It would probably take us, 1 to 2 years. Is this a priority? I don't know. So we need to balance where we actually invest our time in defining the function of these particular types of drugs. And just a toy experiment, a once-off here. Well, we've reproduced it, but looking at atovaquone, which is not thought to have much of an effect on gametocytes. In fact, even though it doesn't, it does still have that same signature that we see with the asexual stage when we look at stage 3-4. So we still see the two hallmark molecules accumulating over the untreated molecule. So in summary, we've been looking at these metabolic perturbations. For now, for whatever reason, they seem to be falling into these broad, these 5 or 6 classes. Of course, the big class needs to be more finely dissected. Probably a lot of interesting biochemistry to be learned from that particular one that didn't give us very strong signals to date. We're trying to use this to look into gametocyte development and think about how we've started analyzing the molecules that have been reported to affect gametocyte development, and they largely fall into digestive vacuole function, which we think is very interesting, but we haven't tested these using metabolomics yet. And we're also trying to understand what that actual profile ultimately means. So in terms of where we're taking this, so we're obviously trying to incorporate both these different types of analytical approaches, looking at other developmental stages. Once we sort of prioritize certain compounds, we can clearly dig deeper into those particular biochemical pathways by doing non-radioactive labeling into particular pathways, for example, using bicarbonate and glucose in the pyrimidine biosynthesis. And of course, eventually, we're going to have to extend this out to see how broadly applicable this is in other genetic backgrounds. With that, I just want to thank the people who have done the work. The ones in red have really contributed significantly and directly to this project. I thank the funders, and I thank you all for your attention. So I think we're gonna do questions, right?
Yeah.
So,
Go ahead.
If anyone has any questions.
Here comes the...
Yeah, please come to the mic, and if you don't mind identifying who you are and where you are.
Hi, I'm Jeff from Boston Children's Hospital. So when you use a single drug and try to go up through a dose range, have you done that for any of them to see whether you get to just catastrophic failure of the parasite, and so do you lose the signal as you get way high above the dose you're trying?
So we really haven't done too many of those experiments. We did it with the atovaquone, which is at the higher doses gave us the same signal, but it's probably not the best drug to use. So a much faster acting drug maybe would give us something very different. And really, we haven't been doing much titration experiments, but we will as we prioritize which ones we want to consider further and really get at mechanism more deeply. Yeah, thanks.
Hi, my name is Mustafa Al-Fawal from UMass Med School. I have a general question. How likely these metabolites has the chemical structure of such metabolite will be modified during extraction, expose them to lab condition versus they were naturally selected to work and that structure will be in a biological condition inside the cell. So when you, by the end of your analysis, you find a structure, how likely is actual the structure in a cell?
Yeah, that's an interesting question. So you're talking about modifications to the drug itself?
The metabolite itself.
The metabolites themselves?
Yes.
I mean, I think the metabolites that we're measuring are definitely true to what's going on within the cell because these are very fast quenching methods that we're using. So I don't expect that there's modifications happening over time, but maybe to elaborate on what I was hinting at, which is that I do think that in some of these experiments, the drugs themselves are actually being modified, and that may be altering what we all downstream may think is the actual mode of action. So anti-metabolites of these drugs could ultimately manifest in what they're actually, how they're interacting with the parasite itself. Yeah, thank you.
Hey, Manuel, great talk. Kasturi Hadar, Notre Dame. So I'm wondering, with respect to the gametocytes and the asexual blood stages, you would actually expect their metabolism to be quite different, right? And I was wondering whether anybody, you or anyone else, has looked at the basic metabolism of the gametocytes, as you go through maturation, and would that be insightful to understanding their development?
Yes.
And so is it just it's published and
No,
and
it's
I
not
don't know
published.
about,
We're
okay.
working on it. It's obviously
Okay.
has other complications because to get well purified gametocytes in large enough quantities to get really good
So what is
signals. As
large
you know,
enough?
we've been
Yeah,
burned in the
but-
past with low signal.
Right.
We're trying to avoid that scenario.
So what's large enough?
Well, we for these experiments we like to use 20 microliters of packed infected red cells, but Darren's approach is what they're doing is on another extreme of sensitivity. So, and the nice thing is you'll see from his talk that we have very complementary data. So there's probably room to, because of my past history, I'm being very cautious about what I call a real signal, and so we've been trying to optimize things to have as much input as possible for these measurements. But yeah, I think we could go much lower, and it's totally feasible. Absolutely.
Like